Okay, well, so let me introduce to Dr. Rodrigo Barderas with great pleasure, who works in Madrid in Spain. And Dr. Rodrigo finished his PhD in 2004 in chemistry. And I had good opportunity to work closely with him when both of us were doing our postdoctorate research at Harvard Medical School in USA. That was during 2006 to 2008, around those years. And we are working on the microarray based technologies. It's really great to see Rodrigo after a long time, and also I'm very keen to know some of the more recent work which he's doing in his lab as a independent PI now at the functional proteomics unit of the chronic disease program in Madrid. So Rodrigo it brings a lot of expertise of microarray based applications. As I mentioned, from long time he has been working in that field, but also he is quite keen into developing new chemistries and developing some newer approaches of doing the microarrays. And here what we are talking is essentially protein microarrays, because our aim is to look at how protein functions can be understood in the miniaturized forms on the microarrays. So that's where I think Dr. Rodrigo's talk is going to be very interesting, when he's going to also give you some flavor of a new technology which is known as phage display based protein arrays. And of course he will cover some aspect of the printing and how to really do quality checks on the protein arrays, and then you can take some of the applications. So on the application side, he has been working actively on the colorectal cancer and other type of cancers, looking at how the biofluids from the cancer patients, especially the plasma sample, serum sample, other kind of biofluids, how well we could screen them on the arrays, and then try to find some potential protein biomarkers which could be used for diagnosis or the prognostic aspects. So with this brief introduction, let me welcome Rodrigo for accepting our invitation to give this particular lecture. And Rodrigo, just for your information, this is a course which is supported by Ministry of Human Resource Development, MHRD, Government of India, under TEQIP program, which is essentially aims to develop the faculty development program of engineering faculty who are working in different parts of India under this TEQIP third scheme. So we have limited number of participants who are all with diverse engineering background, and we are trying to give them the overview of how different uh, type of applications of biology are so much engineering governed. And finally, we would like to take them to journey of big data analysis. So Rodrigo, it's all yours now. You can share your screen and talk to the participants.
Okay. Hello everyone. Everyone can hear me properly, and I don't see if everyone can see. Okay. This is my first time with Microsoft Teams, so sorry because I haven't been unable to put the background. Thank you so much for the invitation. It's a great pleasure to be here. It's a virtual, but I'm really happy to be here in this virtual meeting. First of all, I hope everyone is okay in these difficult situations, and all for his family. So, as Professor Srivastava has told you, I'm going to speak about what we are doing in the lab using different approaches with protein microarrays, from the construction to the application. I'm gonna give an introduction of how to get a protein microarray, and also how they can be applied to different applications. So microarray is a multiplex technology. It consists of an array series of microscopic spots, 100, 400 microns in length, each containing different probes and controls at picomolar concentrations. And then the probe target aggregation is performed in the surface of the microarray to determine the relative abundance of the target. Since an array, just take home message, an array containing thousands of different probes, up to 60,000. Microarray experiments using multiple different arrays, 100,000 different arrays, can accomplish many tests in parallel and dramatically can accelerate different types of research. First, to build a microarray, we have to start from an array library that it can be trivial glycerol stocks, DNA, cDNA, proteins, antibodies, in 96 or 384 well plates. The next step is to consolidate them into 384 well plates, and then to put them in the printer to get the clones printed in the microarrays as micron spots. To get the microarrays, we can use different pins here in the robot. They can be solid or pins with reservoir. There are now piezoelectric microarrays that they are like a printer, a normal printer. It's a laser, so they can deposit small drops, just nanoliters or picoliters of the different drops in the surface of the array with high rate of reproducibility. But usually, if you use solid pins or reservoir pins, it's recommendable to use reservoir because it produces a more homogeneous protein microarray. Regarding the substrate that we can use for producing the protein microarrays, there are multiple surfaces. They have recommended for different applications, polyl-lysine, aldehyde, nickel-coated surface. But for protein microarrays, the nitrocellulose membrane is the most recommended because usually it performs that the three-dimensional structure of the protein remains in the surface of the protein microarray, as well as hydrogens. This is an example of the different performance of different slide substrates for printing of our custom protein microarrays. This is polyl-lysine, hydrogel, epoxy, and nitrocellulose microarrays. These are the treatment that the crystal process, and as you can see, the difference between them are really strong. For example, for polyl-lysine and hydrogel, the spots are not homogeneous, and even the polyl-lysine is not recommended for printing of proteins, at least in our conditions. In contrast, epoxy, it produces a good immobilizations, but nitrocellulose is the best surface for the printing of proteins, maintaining also the three-dimensional structure. So this is an example of the different surfaces that have needs to be tested to be sure that our printing conditions are the best for our custom protein. As this is a virtual meeting, I'm going to show you a small videos of that show you the different treatments that the arrays can have. This is the nitrocellulose microarray. This is a polypropylene microarray that it's equivalent to ELISA treatments, and also you can have here the usual microarray, the usual substrate for printing of multiple different probes that it's recommended for cDNA. For the consolidation of the library, here I'm showing you how we can get the consolidation of a library of pages. We start with the Petri plate with the different pages colonies that has to be manually picked with a robot in a 96 well plate containing PBS and 2.1%, that is our recommended buffer for printing. And then after having the collection in 96 well plates, we consolidate them in these 384 plates using pipettes, multi-well pipettes or specifically we use robots for transferring the 96 well collections to H384 well plates. And for printing, we use for our custom arrays pins with reservoir. It's a small reservoir. And you can see here in the robot, in the platform, the slides are superposed, and you can see here these small devices that produce a vacuum that avoids any movement of the array during the print. Here we put our consolidation library, consolidated library in the field of plates that is going to be picked with the pins, with the, in this case, 88 different pins with the reservoir to be printed, to print our collection in the surface of the protein micro. Okay. Another point to consider when we are working or when we are using microarrays is the detection system to determine the relative abundance of the target. There are multiple different detection systems: chemiluminescence, radioactivity, silver, but the most widely used is fluorescent because we can use 2 or more lasers for excitations of the fluorophores in the surface of the protein microarray. The laser and the confocal scanner collect sharply defined optical sections, and then we can collect three-dimensional images that can be created. Usually two-color scan in less than 10 minutes for the whole surface of the microarray at a high resolutions, about 10 microns, but we can also get the images at 5 micron pixel size. This is a typical sample of protein microarray image. This is the image scan at 532 wavelength, the CY3, and this is the image obtained with the CY5 laser, the 635 wavelength laser. The overlay, it produces an image that usually contains 3 colors, red, yellow, and green. The red, it induces that in the perturbed situations, we have an induced expression, gene expression or protein expression. If the protein remains unaltered in the control and perturbed, it will appear in yellow. And if it's repressed in the perturbed situations, it will appear in green. Type of study that we can get with antibody, protein, and phage microarray. We can. It will depend on what kind of molecule we print in the array. If we print antibody, antigens, aptamers, or allergens, and incubate the array with serum proofs, cell lysate, or living cells, we can collect results related to protein expression levels, protein profiling, or diagnostics. For example, if we print allergens, we can, and incubate with a serum from an allergic patient, we can determine exactly which allergens are inducing in the patient the allergy. If we print proteins, peptides, and incubate with protein proofs, nucleic acid proofs, drug, or enzymes, we can get information related to protein binding properties, pathway binding, drug discovery, or post-translational molecules. Regarding the different type of microarrays, we can get DNA microarrays, aptamer arrays, small molecule arrays. But that's, well, my areas of expertise is related to proteomics, and the course is about omics technologies. We are going to speak to protein microarrays that these arrays involve any kind of protein, antibody, phage, recombinant protein, like peptides, or any complex. Regarding to antibody microarrays, we can obtain information that can be quantitative using sandwich immunoassays or antigen capture immunoassays. In a sandwich situations, we can bind the protein of interest and detect with a second antibody. And in the antigen capture, the proteins are labeled with a fluorophore that can be detected with a laser in the scan. This is the situation that we usually think about when we are using protein microarray. But you can have also in mind that if we work with a protein that will form part of a complex, we can also have these situations. The antibody captures our antigen of interest, but in the complex with all the other proteins that form part of the complex. This is just to have this message at home, because this situation is usually avoided. Usually the people does not consider this situation. How antibody microarrays works? We always have a reference situation, can be protein extract from a cell, and the situation that we can compare with the reference. In this case, a test, for example, the same cells stimulated with repeat. After obtaining the cell lysate and quantifying the amount of proteins, we label each situation with a different word. It's usually recommended with antibody microarrays. We label with different fluorophores to perform also a switch situation to label the reference with CY3 and CY5 and compare with the test with the opposite label. After labeling of the proteins, they are mixed equally, gel filtrated to remove any free time, concentrate, and apply to the surface of the protein microarray, in this case an antibody microarray. So in this case, if we get a green spot after scanning, the protein is down-regulated in the test situation. If it's an equal amount, it will appear in yellow, and if it's upregulated, it will appear in red color. Antibody microarrays can be used to quantify difference between two states in cells, tissue, sera, and almost any kind of biofluid, tear, urine, plasma, sera, etc. And their applications, they have been multiple, just imagine what you can compare with antibody microarrays. The first applications were related to serum profiling. This study was one of the first ones, and it was related to the comparison between the sera of human prostate cancer patients and control. Using 184 unique antibodies printed in nitrocellulose microarrays, the authors were able to separate pretty well prostate cancers in red from control. And 8 antibodies demonstrate a high reliability to discriminate between controls and placebos. Just to acknowledge that this kind of antibody and protein microarrays were developed early in the 2000s. Another application is related to determine difference between normal and pathological states. This was one of the first studies using this technology developed in different labs, mainly by Brian Hub. And in this work, using commercial antibody microarrays, using 226 unique antibodies, the authors were able to find 46 differentially expressed proteins in colorectal cancer in comparison to bare normal tissue, and validating some of the results using tissue microarrays. Other applications are related to drug or radiation effect, that this scheme is related to this application. And the first study using analyzing these situations was performed by Arul Chinayan in the early 2000s in Detroit, Michigan, using antibodies related mainly to the cell cycle. And the authors were able to find difference between normal cells and irradiated cells, and validating were able to validate some of the results by Western companies. But we can apply the antibody microarrays to any other application, to response to treatment, effective drugs, etc. Regarding to protein microarrays, the term is usually related to natural proteins or tumoral proteins, recombinant open-reading frames expressed in bacteria, insect cells, or also related to phages. In these situations, we printed the protein, the phage, or even cDNA in the case of NAPA, to use the protein microarray to detect direct interactions or protein-protein interactions, small molecule, or kinase studies. How we work with this kind of protein microarrays? We have to compare different situations, control versus treated, use usually one array per condition. In this case, the array is incubated with serum and the presence of antibodies specific to a situation or disease situation. It can be detected using a secondary fluorescent antibody labeled with fluorescence. The image is collected in a microarray scanning, fluorescent microarray scanning, and after that analysis and the comparison of controls and the disease situations, we can get those proteins specific of the disease situations by bioinformatics. Applications, multiple again, for the detection of protein-protein interactions, ligand-specific binding, detection of substrates of phosphorylation of kinases, and the most important applications have been related to the detection of immune response in autoimmune, cancer, and more recently to neurodegeneration and neurodegenerative disease. The first reports using this technology were also in the early 2000s by the group of Michael Snyder in the Stanford, and they get a yeast protein chip with about 90-95% of the whole proteome of the yeast. They use these protein microarrays to identify proteins able to bind to nucleic acids, lipids, or small organic compounds, or even for protein-protein interactions. In this image, I'm showing you the results related to the identification of calmodulin-binding proteins just by the incubation with calmodulin labeled with violet. And as you can see, the authors were able to detect those proteins able to bind to calmodulin by using a streptavidin labeled with a secondary fluorophore. Also, the authors were able to detect the specific calmodulin binding site of the newly identified proteins that were able to bind to calmodulin. The authors also used these proteome yeast arrays to detect substrate of phosphorylation. The authors used the arrays to incubate the proteome arrays with purified kinases, incubated with radioactive phosphorylated ATP. After incubation, the author was incubated with a film to detect the radioactivity and identify new substrates or protein substrates of kinase. But the main application of protein microarrays have been related to identification of autoantibody profiles, because and mainly in cancer, because cancer patients spontaneously produce autoantibodies against tumor antigens, proteins that are usually not encountered by the immune systems, because they are usually overexpressed. They suffer from alternate modifications, for example, alternate glycosylations, or they are continuously degraded or overactivated or mutated. So these autoantibodies against these altered cell proteins can predate or anticipate cancer by years. So this has produced a really whole attention whole bunch of reports to identify these autoantibodies and the target proteins. One of the first reports was related to the use of about half of the protein array developed by in vitro gene. They were commercial, but now they are discontinued. And the authors, including me in the authors, we were able to identify protein targets of autoantibodies against 43 different autoantibodies that were able to discriminate between healthy situation and colorectal cancer. The seroreactivity, the differential seroreactivity found in the microarrays is shown here in the slide. And you can see the differential intensity between the tumor group and the control group for 6 selected proteins. The authors were able to identify also a predictor for colorectal cancer diagnosis, which also patented. Other study related to the use of these protein microarrays and cell profiling was this study published in Journal Protein Research that combined serum and tissue profiling in bladder cancer, and where the authors were able to distinguish 2 proteins that have differential, or were able to differentiate the patients according to the survival. The other type of protein microarray that I wanted to present you is NAPA, nucleic acid programmable protein array. In this case, the situation is different. The proteins are per se not printed in the array. They are printed as cDNA. The proteins are cloned in the cDNA of interest, tagged with a GST tag usually, and the cDNA is printed also with an antibody anti-GST. After printing of the DNA with the antibody anti-GST, the protein array is activated using cell-free expression system. So the target protein is produced in the surface of the array, captured with the antibody, and then the array can be used for all these applications that I have told you before. For protein-protein interaction, enzyme substrate identification, or identification of complexes. This is an example of the performance of the nucleic acid programmable protein arrays in NAPA, and the advantage, the main advantage is the absence of expression and purification of the protein separately to the array. That avoids problems related to the stability of the proteins and degradation issues. The expression in a mammalian milieu, because the cell-free that is used to activate the array is a reticulocyte or HeLa cell lysate. So the expression in mammalian milieu assures a natural folding. The proteins are produced just in time for assay, and they are expressed and captured in the circle of the array an amount of 700 micrograms, median, that it's about 1,000-fold more than protein spotting arrays. And the other advantage is that the arrays are stable on range until activated, in contrast to protein microarrays that need to be stored usually at -20 or -18 for prior to the use in the living applications. And the last protein microarray I wanted to present you here is the teacher and phage microarray that we have been using in the last year. It consists of the integration of 2 high-throughput technologies. On the one hand, phage display, and also the protein. The first study using this technology was also developed or was reported by Richie Young in Michigan. It's a prolific author in the field of proteomics genomics in cancer. And as you can see here, after printing and scanning, it was observed a differential seroreactivity between prostate cancer and control. Using 1,000 different sera from prostate cancer and control, authors were able to identify a set of about 20 different phages that were able to discriminate between prostate cancer and control cancer with a high performance. The raw cures were considerably better than the antigen used for diagnosis of prostate cancer, the PSA. But why is so much interest in identifying autoantibodies and the protein targets? During the cancer formation and progression, all these genetic alterations that are produced in the tumor are translated into alterations in proteins. These alterations in proteins can induce antibody production, so the antibodies and the target proteins can be subsequently identified using protein microarrays, thus comparing controls and cancer samples. How we work for serum screening with phage microarrays. As well as with protein microarrays, we have two different cohorts of patients, controls, and in this case, colorectal cancer. We incubate each array in separate with any sample, and after detection of the presence of autoantibodies using a secondary anti-human anti-IgG antibody, we can detect the presence of these autoantibodies in the target proteins with the microarray scanning. After that analysis, comparing the control group and the cancer patient information, we identify the specific proteins target of autoantibodies in these diseases. And how we work with this methodology with phage collection. First, depending on our disease, we, in this case, colorectal cancer, we isolate the tumor. From the tumor, we isolate the total mRNA, clone the cDNA to obtain a cDNA library that is inserted in the T-cell vector, that is the genome of the T-cell phage. And after in vitro packaging, the phages display in the surface specific peptides or proteins of the tumor that carry all of the alterations produced in the tumor, mutations, aberrations, frenzy, etc. Here, in the library of phages, we depict in red the specific colorectal cancer phages that can be enriched in different rounds of selections simply by incubating with control sera to remove non-specific phages in blue, and then enrich in specific phages in red by incubating with colorectal cancer tumoral sera, the IgG from the sera. After these selections and infection in Ebola and amplifications, we can enrich in each round of selections between 100 and 1,000 times the collection, the original library, in phages displaying peptides specific of the pathogen. By simply repeating this process 3 or 4 rounds, we can increase the original library. We can enrich the original library in 1 million or 10 million in phages displaying peptides specific of our disease. Prior to print in the arrays, we have to demonstrate or to identify which surface is the best option for the printing of our custom array. This is the image that I showed you at the beginning of my presentation, and you can see that the nitrocellulose was the optimal surface for printing of this page. Then, after monoclonal page selections, we printed 2,000 pages and controls, monoclonals, and in nitrocellulose protein microarrays to detect the presence of the T-cell pages with antibody anti the T-cell phage and the specific peptides specific of our disease using an anti-human IgG conjugated with alexafuloxin. This is the quality control of these arrays printed with the phages, and as you can see in the image, the array containing 2,000 phages and controls in duplicate was really homogeneous in intensity of the presence of the phages, and also with the secondary antibody, the anti-human IgG conjugated with Alexa 647, we observe the presence of a low value. Also, it's important when you get an array, you have to demonstrate good reproducibility between the different bytes, because it's not the same usually. The fair array printed in the robot that if we have a collection of 3 different arrays, if you compare the first one with the last one, maybe it can be some problems of reproducibility. So you have to demonstrate the arrays show a good reproducibility by intra-array reproducibility and inter-array reproducibility. The intra-array reproducibility was also was very high, near 0.97 squared, and just comparing the spots printed in Nucleic, and the inter-array reproducibility was higher than 90%. So in these situations, the next step of this methodology, 57 pages, is use screen the arrays with normal tumoral samples to demonstrate the presence of a differential cellular reactivity between the groups. Here, comparing the tumoral samples and the control sera, we observe this differential cellular reactivity in these spots near green or near yellow in comparison with the total green observed in 50%. The last step of this methodology is the data analysis, identification of the specific phases, and verification of the differential cellular reactivity using orthogonal technique. In this report, we observe 78 different phases having a differential cellular reactivity between tumoral samples and control samples. After amplifying these phases and sequence, we observe the presence of 43 unique sequences, selecting 6 of them for further validation. We observe here the differential cellular reactivity observed in the microarrays, in the tumoral group, and in the control group for the 6 selected phases. For validation of this specific cellular reactivity and to demonstrate if they were or they possess some diagnostic ability of colorectal cancer, we proceed to validate this cellular reactivity by other techniques. We use ELISA and a set of sera different from the ones used in the protein microarray. We use a collection of 50 serum samples for healthy controls, 50 from colorectal cancer in different stages of the pathology, and 53 from other cancer and gastrointestinal diseases to demonstrate the specificity for colorectal cancer. Here you can observe that the differential cellular reactivity was also observed by ELISA in comparison to 2 different proteins, Annexin-4 and CA, that didn't possess any differential cellular reactivity. We also calculate the specificity and sensitivity of the test, having into account that we have a perfect precision. The area under the cube should be 1, good precision 0.75, and no precision 0.5. And we observe that our potential phases with diagnostic ability, the combination of them possess an area under the cube near 100% of sensitivity, 100% of efficiency, and a sensitivity and specificity near 90%. In early stages, the reliability of the test and the sensitivity and specificity was also very high, and also in comparison with the carcinoembryonic antigen, that is the marker used in clinics for colorectal cancer. So our best combinations was able to detect also colorectal cancer with good performance and colorectal cancer error. So just as a brief summary of the talk and those messages to take home, just custom and commercially available antibody pages and protein microarrays are robust techniques for protein studies with a high degree of reproducibility. Actually, there are commercially available surfaces for antibody and protein microarrays from different companies ready to be used. However, they are really expensive, and custom protein microarrays offer the same reproducibility as those commercial ones, and they are a cheaper alternative for proteomic studies. The main message for you is that PHADES, antibody, and protein microarrays can be used for many different applications, including autoantibodies, autoantibody identification as biomarker of diseases, analysis of phosphorylations, protein-protein interactions, epitope mapping, and that protein microarrays-based proteomics is so versatile and flexible that can be useful for any area of research. Just can imagine what you can do with this technology in your area of research and apply it. And these are the main message of my presentations. Thank you so much for your attention. If you have any questions, you can contact me at my email address, and also here with I'm happy to answer all any question that you have.
All right. Thank you so much, Rodrigo, for giving a very elaborate presentation. Talking about different nut and bolts of microarray and how many type of microarrays are possible. Then showing finally the potential applications on those arrays. So, given the wide spectrum of the diversity what we have in the participants, I think it was probably good for everybody to get a flavor of that. So let me ask participants if they have any question, if they want to learn something about any aspect of microarrays. We are open for any discussion or questions. I think it's a good idea for participants to now start opening up and talk to us. Argo, you have any question on the arrays platform with Dr. Rodrigo?
Hello. Yes, sir, I have a question. When you are printing a duplicate spots in the microarray chip, we are seeing that whenever we are including an antibody, like when we are incubating it with the antibody, the intensity application, like whatever the intensity comes out, is not same or not identical. However, we take it with the statistical exception that it should be less than a standard deviation of 20% or 25%. Now, my question is that if I am incubating with the same amount of protein, or say, and the spotting is also done accurately and exactly, why this biasness is there? And always we have to rule out statistically, means after the intensity recordings have been done, like the interchip variability, intra-chip variability, which is creating a, what to say, like a troublesome for the post analysis, post microarray analysis, and how this can be salvaged.
Well, we usually
many aspects. I think maybe Rodrigo can break it into small parts and then answer them.
I mean, yeah, the problem with duplicates is, I mean, it exists. What we have observed is the duplicates. I mean, the location of the duplicates is better to have near each other. Our duplicates are usually printed, I mean, directly in the same position of the microarray, not in a different position. Just to try to avoid this troublesome, because sometimes in the protein microarray you have small deviations in the incubations. The incubation is not 100% equal in the whole surface of the microarray, so it's better to have the duplicates near each other. And with these situations, at least this troublesome for the incubation, I mean, you can avoid this part. Regarding other issues, I mean, for the post-analysis, we always work with the median of the duplicates, or if we print triplicates or quadruplicates, we work with the median intensity of the replicates to avoid this situation, because even just by chance, you can get one spot having higher intensity than the real one. So working with the median, you can solve some of these problems. I don't know if I have answered really well the question. If you want, you can write me, and I will extend more in my answer, because the
Right. I think,
bioinformatics analysis is
right. I think broadly you have covered, because many of these things are also linked to the analysis aspect eventually, when
yeah.
you see the issues with duplicate spot and all. And that actually brings to question of another participant, Dr. Krishna Gopal. He is asking, is there any image processing method which is employed for analyzing microarray images for the disease identification? So would you like to share some insight with him?
Me, I mean, if it's he's a bioinformatician, I mean, I mean, his the algorithms that they use sometimes are quite difficult to understand for even for me, and they are he's going to bring us more knowledge on this problem of the normalization in the protein microarrays that sometimes is a quite difficult problem. When we work with the normalization of the protein microarrays and the normalization between the duplicate replicate, we always work with the median and also with the median of the intensity of the different arrays. Because if we have two channels, in the case of T7 phages, we have a red channel and a green channel. The green channel is going to be always, it has to be always the same. So the median intensity has to be the same in all arrays. So we can work with this median intensity of the T7 phages to normalize the microarrays and get good performance and good results at the end of the data analysis.
Right, great. So I think, as I think this question was quite relevant, most of the time, I think Krishna Gopal and others, generating data is not very difficult, because once you have the, availability of these arrays and contents, you can generate big data very easily. But then, after exporting the data, so actually there is a lot of things goes on. When you have a microarray slide, you have to put the grid properly so that all the featured data could be extracted out. And then from there, you want to export the data, and then you want to normalize the data. And then you want to make sure that, what you are getting is going to make any biological sense from the statistics point of view. And that's why I think in this course, we also have involved a professor from the biostatistics, mathematics department, who's going to talk about different concepts of normalization and looking at how one could make sense of this data set. And eventually, whether you get big data from microarray or mass spectrometer or any other platform, there are going to be different algorithms which has to be employed for making sense of the data finally. And that's where more of the machine learning approaches are coming forward. So I think all of these will be covered in the course as we go along. Given it is first day, I'm sure things are a little complex for many of you to follow up. But very soon we'll try to fill the dots and try to show you the workflows. In fact, we will also give you a one hands-on session where you will get an idea that how to take the microarray images, extract the data with the GPR file, and then try to take out those values, and then try to plot those and see your own way of analysis. It's still what you may realize, many of you who are good into software part, that is still there are not very good softwares available which could do immediately the whole analysis, in an automated manner. So many things we have to do in a different steps with different software, and some of them are open access, some of them sometimes become commercial. So it's very difficult to really utilize same workflow by all the laboratories. So people do try different things as per availability of the commercial version of software or the open access availability. So we will take these things more as we go along in the course. Any other comments or questions? Arga, you have something? You asked
Yeah,
median of.
So,
I
like
just
what Rodrigo is suggesting, Professor Rodrigo is suggesting, you are generally taking median of two spots. First, you
yeah,
are taking median of the two replicate spots. Once the
well,
median has been taken, you will get a feature. Now, among the intra chips for that particular protein, we are also doing a median normalization.
But
Okay.
you mean median normalization of the replicate or between
Median
different arrays?
yeah, median normalization of the duplicates as well as the median normalization across the chip. This is the because we sometimes found that the quantile normalization seems to be promising. And one more thing, according to you, like what are the condition of excluding two spots? Like you will not consider only if you are finding this kind of features in the array chip.
Well, I mean, sometimes it's easy to see which spot is the one that has to be removed, because I mean, in the grid, in the protein array, if you have the duplicates, if one of them has a signal intensity in the edge of the spot, this intensity is irrelevant, so you have to discard these spots, or even if it's a comet or has a donut, the spot that sometimes it can happen, you can see which one should be removed from the posterior analysis.
And like the negative values, in some cases we can find some negative values also. Like
Yeah.
are you dealing with some that like making it positive or bringing it to some baseline?
Well, I mean, the negative ones at the end is because the background outside of the spots is bigger than the signal inside of the spots. So this usually makes those spots irrelevant for further analysis. In case you are comparing, I mean, in our case is for set of proteomic analysis, comparing control and colorectal cancer or neurodegeneration. So we prefer to work with those spots having higher intensity in our disease than in the control sequencer. So we don't have any problem with the absence of signal, because sometimes it can be a problem during the printing. Also, I mean, just giving some extra information about data analysis. We also, after extracting the information from the array, the GPR files and the data analysis, what we usually perform is to use 3 different algorithms. There are multiple platforms, and in different laboratories they use different algorithms. We prefer to use friendly and web-based platforms like Babelomics, Pomelo, multi-experiment viewer, and we usually combine 3 different algorithms to get extra information, because sometimes with using just one algorithm, you get 3, 4 proteins, and using another one, just 1 or 2 proteins are the same with this algorithm. So you can increase the number of reactive proteins, and after that you only have to validate the results. But it's better to work with different algorithms for the data analysis than using just one.
And I guess, on the little bit more on the clinical and biological aspect, whenever you are doing analysis after that, the good strategy is to make a smaller array from the identified list of the significant proteins, and then, make the smaller validation array on which you can again do the experiment and see how many of your stats are still, holding up. And then, of course, finally, you have to go for the blinded validation in the pipeline of biomarker discovery. So none of these can be taken immediately forward because of, the constraints of data analytics, the way we do in different software. So that's where different level of validation experimentally is required. But this is how we funnel down from the, 40,000 to 1 million spots, and narrowing down to maybe, 200 spots or 400 spots, and try to then come down to 5 potential biomarker, which is not easy to get in, and that's where a lot of challenges are required. So I know Dr. Rodrigo has to leave for another meeting, and I appreciate that he stayed so long and gave some insight. So thank you so much, Rodrigo, on behalf of
Thank you so much for the invitation.
the TEQIP program, and we are glad that you spent some time with us and shared your research.